Thank you. I'm very excited to be here. I have to say that I might be the outlier in this session, consider we actually don't do any sensor work. So if that's what you're looking for, this is time to leave. But don't. So for this audience, I don't think I need to spend too much time on this slide. As we all know, there is a need to improve the productivity of drug development, and to do so would require us to develop more accurate preclinical human-based tissue models to better predict the outcome of clinical trials, and hence to reduce the attrition rate at a later stage of drug discovery and bending this productivity curve. Now, this community came up with a variety of approach to this problem, and they are all very promising. And this is a highly interdisciplinary area of research that integrates organoids, bioreactor design, microfluidics, right, 3D bioprinting, and tissue engineering strategies. So the question is, what motivates us to develop our platforms? So what kind of feature do we want in our system? First of all, we really want our platform, our model, to be relatively high throughput so that we can screen hundreds of experimental conditions robustly and cheaply. Many biological processes are very complex. Some biological effects are very subtle, so we really think you need a reasonable experimental throughput to uncover some of these biological processes and effects. And we also want our platform to have this open well design so we can easily extract the tissue that we built from the system for downstream analysis, like histology or gene sequencing. We basically want to be able to treat the tissue we built like a real human tissue biopsies. And we also want to be able to incorporate these multi-layered tissue structures that includes different type of cells, and the cells should be able to freely move within the tissue, within the matrix, without any hindrance from any synthetic materials. And we also want to be able to incorporate tissue-specific topographies into our model so that we can look at these cell-specific niche that are important to different tissue types. And lastly, we want to be able to apply mechanical actuation on our tissue, as we all know that mechanical forces, and this includes vascular perfusion, fluid flow, can have a significant impact on tissue functions. So we want to be able to incorporate all of these features in our model system, and for that reason, we developed this two platform called iFlow plate and Angel plate. So how does this work? The design of these two platforms are actually very, very simple. It's probably as simple as it gets. All we did here is we took a standard 384 well plate, we patterned two channels to connect three wells together to form a single unit. And so on a plate, you can get 128 of these independent individual units. And then you take a hydrogel, you cast it into the middle well here. You can seed cells into the hydrogel, or you can place the cell on the surface of a hydrogel, or you can do both. And the cell will self-assemble within the matrix and forming that tissue. Now to perfuse that tissue, what we do is just simply rock the plate back and forth. Using gravity-driven flow, you can perfuse cultural media from the first well, through the porous hydrogel and through your tissue, and it goes to the next well and back and forth. Now, if you want a little bit more control over the structure of that tissue you built, you can upgrade this to the Angel plate, which contains a patterned sacrificial material inside the well. And then if you cast a hydrogel over the sacrificial material, remove the sacrificial material, that will leave behind a perfusible network, which you can then use to guide the assembly of the cells into a functional tissue with more controlled structures. So very simple strategy. And then using these two type of plates, we were able to develop a broad range of tissue models that includes the blood vessel, vascularized liver, placenta, lung airway, alveolar model, kidney proximal tubule, and colon. So in this talk, I'm going to specifically focus on the blood vessel model, the colon model, and at the end, I'm going to just talk a little bit about our lung model. So first, the blood vessel. So how do we build a blood vessel model using this platform? And in this case, we are specifically want to create a perfusible, self-assembled microvasculature. And to do this, we used a fibrin hydrogel. As we all know, fibrin, indocylindole really likes fibrin. And then what we do is you just simply take the indocylindole embedded in the fibrin matrix. The indocylindole here are labeled in GFP. They are fluorescently green. And over time, these indocylindole will self-assemble into a vascular network. On day 5, they will form this continuous network that then connects to the inlet and outlet channel and become perfusible. To visualize this process, we added a color dye, a fluorescently red colored dextran dye in the inlet well. And you can see on day 1 and day 3, before the network fully establishes, it's not very perfusible. The dye have a hard time going through this porous gel, but once the network is fully connected, then the dye can easily go from inlet to the outlet. And then once the connection forms, the network becomes fairly stable, and we can maintain this for at least 11 more days beyond this point. Now, because this is based on self-assembly, the endothelial cells, they can wrap around themselves or around each other to form these microvessels with these open lumens. But you do get this broad range of diameters, and because of that, there is a broad range of shear stress that the cells will experience. And here, just to show you, within the first 5 days, during this remodeling process, if you look at the vessel closely, you can see some of these modeling process, like the fusion of 2 vessels, splitting of vessels, sprouting and vessel regression. But by day 5 and 7, the structure becomes fairly stable unless you treat it with a drug. So here we want to show that not only we can create these tissue manually, but we can automate this entire process using a robotic liquid handling system. And here you can see that the perfusible vessel made here is very similar to the vessels that we casted manually. And you can scale this up to create this array of vessel tissues, and this is what they look like. You can use dextran dye to check for their perfusibility, and you get this array of perfusible vessels. But from the image, you can already see that success rate is not 100% because this process is based on self-assembly, which is a little bit stochastic. There's a little bit of randomness to it, so the vessel don't always connect to the inlet and outlet channels. So some vessels are not, not all of them will be perfusible. And because of that, we do introduce a quality control step into our experiment. So what we do is, when we make these tissues, we then first check for their perfusibility by adding the dextran dye before we do any drug treatment or any experiment. And then we take the perfusible vessels, also check for their vascular structures, and then we just distribute them under different experimental conditions that we want to test. So in this case, we want to check this chemotherapeutic drug at a different dosage. And this is the vessel before we apply the drug treatment, and then after we treat the vessels for 3 days, 72 hours, and then we add the dextran dye again to check for perfusibility, and that's what you see. So from this experiment, we can extract a number of information. One, the endothelial cells, they are GFP labeled. So if their viability goes down, then their GFP signal goes down. And so that's exactly what we see at a higher dosage. We see this nice dose response curve, right, where the viability, the GFP signal comes down. And because we are perfusing the vessel with these dextran dye, we can also check for their perfusibility and permeability. Again, we're seeing the same trend at a higher dosage. The network becomes significantly disrupted, and so the number of non-perfusible vessels also goes up at a higher dosage. And for the perfusible vessel, we can then check for their permeability, and what you can see here is some of the vessels that are perfusible are already starting to leak, right, have an increased permeability at these even lower dosages. And then from the images, we can analyze the structure of these vessels. So here we look at about 9 different structural parameters, and to visualize that data holistically, we did this principal component analysis. And what you see here is that if we look at the structure of the vessel, for most of the lower dosage treatments, the structure of vessel hasn't changed very much, and they cluster around the no treatment control. But for the highest dosage, 1 micromolar of this drug, the structure has been significantly damaged, and it clustered away from the no treatment controls, right? So it shows a significant difference. But what we can learn from this is that it seems permeability measurement, at least for this particular drug, is even more sensitive than structural analysis or viability analysis on uncovering these toxicity effect at these lower dosage values. Now, there are some limitations with this kind of self-assembled vascular models. One is that there is structural variation from well to well. That's something that's a little bit difficult to control. But if you care about this structural remodeling, right, with your drug treatment, then this is the model you should definitely consider. But if that's not something you care, then we can actually further simplify these blood vessel model to turn them into a single tubular vessel, so very even simpler structures, right? And to do this, we use this angioplate that contains a pattern sacrificial material inside the well. And then we can create these microchannels inside the hydrogel. We can populate that channel with endothelial cells and surrounding that vessel with different stromal cells, so you get this tubular vessel. And in this case, we really wanted to look at the interaction of different stromal cells with these blood vessels. So specifically, we looked at fibroblasts, parasites, and a combination of fibroblasts plus parasites. And what we found is that the presence of these stromal cells, fibroblasts, parasites, can significantly improve the barrier function of these vessels, and the combination of fibroblasts, parasites is also good. Okay. Now, one key advantage of this model system is that, so before when we're using the IFO plate with the self-assembled vasculature, if we have a 10 million endothelial cells, we can maybe cast a column of tissues that's about 16 tissues. But now because we have a simplified structure, now with 10 million endothelial cells, we can pretty much seed an entire plate of tissues. So that's over 100 tissues. So we can really scale this up with high reproducibility. So here you're looking at, so we measure the permeability of these tubular vessels, and you can see there's a very small variation between these vessels within the same plate, also from plate to plate, between different plates. And then we can scale this up to the entire plate. So using this, we look at 10 different chemotherapeutic drugs at 5 different dosages, and we were able to generate this kind of dose response curve for these chemotherapeutic drug, and you can clearly pick out the vascular toxic drug, this one here, this one, and these two here, okay, just by looking at the dose response curve. Now, we also collected the cultural media and analyzed for cytokine secretion profile from these tissues. And so in this case, we looked at about 19 different inflammatory cytokines, and again, to visualize that data holistically, we did principal component analysis, and what it shows that for most of the non-toxic drug, they have an inflammatory cytokine profile that is very similar to the no treatment control. But for these two drugs, bortezomib and vincristine, they show a very, at this high dosage, they show a very different inflammatory cytokine release profile. Right? Now, what's really interesting is that if you just look at the permeability changes, you know that these two drugs are toxic, but by looking at the cytokine profile, you can see they are toxic in two very different ways because their cytokine profile don't cluster together, right? They are very different. So in summary, what this shows is using this angioplate blood vessel model, we can do this type of drug screening, at least looking at vascular toxicity. So now I'm going to switch gear a little bit to talk about the colon model we have. So on one hand, we know that we can create these self-assembled perfusible microvasculature using iFlow plate. And on the other hand, based on using literature protocols, we can grow these colon organoids derived from primary colon stem cells. And so what we want to do here is to combine the two, right, to create these vascularized colon organoid. And this would require us to optimize the matrix as well as the media formulation. And so here I'm just going to talk a little bit about the matrix optimization. So we all know that endothelial cells really likes fibrin, but we also know that organoid, colon organoids, they really need matrigel. So we need some sort of compromise here. And what we found is that 90% fibrin plus 10% matrix gel give us the best matrix that can support both type of tissues. And so with that matrix formulation, we were able to create these vascularized colon organoids, and we show that the endothelial cell, again, they are able to self-assemble, become perfusible on day 5, and their structure is maintained after that, while the colon organoid continues to grow over time along with the vasculatures. Now, one nice thing about this platform with its open well design is we can easily extract the tissue from the device and perform histological section on the tissue and compare it directly with native colon tissues. And so that's what you see here. From the histological section, you can clearly see the colon epithelium cells are maturing and polarizing with Villian staining on the apical surface, while the C31 endothelial cells, they self-assemble and form this perfusible vasculature that closely wrap around these colon organoids. And this is exactly what you see in the native colon, which is also highly vascularized. The blood vessel in the native colon are also in close proximity to the colon epithelium cells, and you don't get that kind of vasculature in the traditional organoid matrix dome culture. So next, we wanted to model colon inflammation. So we treated this tissue with TNF-alpha, an inflammatory cytokine. And the first step of this inflammatory response is the activation of these vasculatures. So when we treated these tissues, the endothelial cells now start to have this increased expression of ICAM1 on the vascular inner luminal surface. And then we perfused circulating immune monocytes through the vasculature. The monocytes are labeled in red color here. And so again, you see the TNF-alpha treated groups where the vasculatures are activated. The red monocytes then, as they go through the vessel, gets trapped in the vessels. And then the next step is the monocyte need to migrate across the vascular barrier and enter the lumen of the colon. And that's what we see here is the red-colored monocytes. They were able to migrate across the vascular barrier, aggregate inside the lumen of these colon organoids that's surrounded by these blood vessels. So what this shows is that we can use this model to model this inflammatory process that involves the recruitment of immune cells through the vascular systems. Okay. Now, again, there are some limitations with this model. One is that the colon organoid has closed lumen, so the luminal space is not really accessible. We cannot really measure the barrier function of these colon epithelium cells. And the other limitation is that the colon organoid, they do form this budding structure, which represents the colon crypts. That is important to the colon tissue, but that formation of these budding structure is highly random, difficult to control. It's very much a stochastic process. So we want to see if we can overcome these two limitations. And so we thought, can we use the iFlow plate in a different way? So instead of seeding the cells inside the gel, what if we seed the cells on the surface of the gel to form this barrier, epithelium barrier, that allow us to access the lumen space and also look at the barrier function. Now, we can definitely create just a flat layer of epithelium cells on the surface of the gel, but if that's the only thing we did, then we will be losing on an opportunity to introduce these crypt topography that's important to colon. So what we did here is we 3D printed a stamp that has this array of pins at the tip, and we can use these pins to poke holes into the hydrogel to create this array of invagination inside the gel. And now, if you see the cells on the surface of that gel, the cells will then follow that surface topography and automatically form these crypt-like structure. And then we can get these array of crypts consistently without having to rely on the stochastic nature of budding of organoids. Now, if we look at these tissue a bit more closely, we can also see that, so here we label the proliferating cells with EDU staining, and you can see the proliferating cells are aggregating at the bottom of these crypt, which is what we want, right? That's the stem cell niche. But this process, it seems, is not just driven by the presence of these topography. It seems it's also at least part of it is driven by the presence of an interstitial flow through the hydrogel that creates this chemical gradient across this colon barrier, because we didn't see this kind of patterning in the traditional static 384 well plate format that doesn't have any perfusion or any chemical gradients. And next, we wanted to incorporate vasculature into this model. So we did exactly what we did before. We added endothelial cell into the hydrogel. They self-assemble, forming this perfusible network that connects to the inlet and outlet well. And they do this at the bottom of the well. Some of these vasculature can even migrate upwards and to wrap themselves around these colon crypts that's on top. Now, because the confocal is difficult to visualize this because of the light scattering, so what we did is we then cut across this tissue and section the colon crypt. And so this is the histological section. You can see the colon epithelium cells, again, are polarizing with Villian staining on the apical side, while you have these CD31 positive vasculatures, microvasculature self-assemble, and they are wrapped very close to the colon crypts here. So you get these truly vascularized colon crypt barriers. And then next, we treated the tissue with TNF-alpha and interferon gamma to stimulate this inflammatory response and to look at different cytokine gradient across this barrier. Now, even before the treatment, what you can see is that just the presence of vasculature can provide a lot of different cytokine gradient across this barrier. Right? Now, during treatment, the inflammatory cytokine goes up. That's not surprising. And after treatment, some of the cytokine gradient actually reversed, some are maintained. But overall, I'll say that the message here is the presence of vasculature is important. They do provide a lot of biochemical signaling and cytokine gradient that are missing in the avascular conditions. So now I'm going to use the last few minutes of my time just to talk a little bit about our lung model. So using the angio plate, we wanted to create an airway tube model and alveolar sac model. So the angio plate is the device that contains this patterned sacrificial material. By controlling the structure of that sacrificial material, we can create different patterns, control the structure of the tissue inside the well. So for the airway tube, we create a channel, we populate that channel with airway cells, and then you get this nice airway tubes. For the alveolar sac model, we create this chamber-like structures and populated that with primary alveolar cells, and then you get this alveolar sac. Now, with some differentiation, we can also get ciliated cells on the airway tube, and we also get type 1 and type 2 alveolar cells on the inside the alveolar sac model. Now, what's interesting here with the airway tube is if you incorporate long fibroblasts in the gel around the airway tube, they can actually further support airway epithelium cells and tighten the barrier of that airway epithelium barrier. Now, what's really cool about this platform is that we can actually link it with an external small animal ventilator. So we can directly ventilate these engineered tissue inside the plate and to apply this mechanical actuation on these tissues. So for example, for the alveolar tissue, because it doesn't have an outlet, it receives all that pressure, so it really expands with this ventilation. Right? And we're not doing this just on a single tissue. We're applying this mechanical actuation on an array of tissues that's inside these well plates. Right? So we can really apply this mechanical actuation on every single tissue. Sorry that the video is not in sync, but in reality, all the tissues are actually actuated at the same time, so they're synchronous. So in the future, we are working on using this technology to look at how mechanical actuation can impact the function of these tissues. So in summary, I hope I have convinced you that using iFlow plate and angio plate, we can do high throughput screening. We have this open well design that allow us to extract the tissue for downstream analysis. We can incorporate multi-layer tissue structures, introducing these tissue-specific topography, and apply mechanical actuation. And lastly, I just want to say that me and two of my students, we do hold equity in the startup company from my lab, Organobiotech, which is actively commercializing some of these plates that I just shown you. So with that in mind, I take everything I told you with a grain of salt because I can be very, very biased. And then lastly, I just want to acknowledge the students who did all the work. So Zhang developed the vasculature model, Shuwansi developed the colon model, Alex and Kimia developed the lung model. So with that, thanks.
Thank you, Dr. Zhang. It was really a nice presentation. We have time for a couple of quick questions, if anyone has.
So there's one question in the back. I pass you the microphone.
Thanks. Hi, Lisa Mojnieks from Elvis's Microfluidic Innovation Center. Thanks for a great talk. I was wondering on your lung model with your angio plate, if you have different parts of the plate which are the airway tubes and different parts which are the alveolar sacs, can you connect those two parts with a little piece of tubing and have a total lung on chip? Thanks.
Yeah, they are not connected right now, so they're completely separate models. But yes, potentially you could connect them, but that the two tissues requires different type of cells, right? The challenge is how to pattern the cell accordingly. We're not there yet, but we are thinking about potentially integrating this with, lung organoids that can spontaneously differentiate into these different type of cells. But that's a separate story.
Thank you.
Okay, there's another question.
Thanks for a great talk. I'm Sanjay from Seoul National University. I have questions regarding the colon models. You showed two colon organoids. And can you compare the two types in terms of the benefits, like in testing some sort of efficacy, is better to use this model compared to other? And can you compare the benefit of two colon organoids model you showed?
Okay, the two type of model, colon model we showed. So one is really building on the other. So in terms of the engineering aspect, the second one I showed with the cryptopography is definitely better. But in that model, we use the KICO2 cells right now. And so we are working on upgrading that with colon organoids, which hopefully will then become will allow us to check all the boxes that we want in these colon model system. And the first one is using the colon organoid, but it has these other limitations I mentioned.
All right, if anyone has any more questions, please catch Dr. Zhang around tea or coffee. And let us thank our speaker again once more.